We are going to prepare the agarose gel now to see that how our plasmid DNA has been isolated. So if we look into the horizontal gel electrophoresis system, it has got a lid and we can remove the lid, and then inside there are two stoppers that we can put actually them onto the gel tray. Aruj, can you do that? So when we pour the liquid, it can drop down. To stop that flow of the liquid, we need these stoppers so that the agarose gel can be solidified inside the gel tray. So she's just pressing and setting this tray into the grooves of this rubber material. So you can see that these stoppers now they won't allow the agarose liquid to flow out of this cassette. And then she also has some combs that need to be inserted into the gel so that we can pour the samples in them. So the well formation will be by these combs, and if you can see that this comb is slightly above the surface of this tray, okay, and these wells will be used for loading of our DNA samples. In order to prepare the gel, what we need is that we need 1% agarose in 1x TAE buffer. So we have taken 50 ml of 1x TAE in the flask for making the gel, and we will now dissolve agarose into this flask. So what agarose we have got is okay, we have got ultra pure agarose from Invitrogen. This is a very high quality agarose, in fact. Okay, and it can be used for making the gel here. We will take half a gram of this into 50 ml, and that will make actually a 1% solution of agarose. So she's going to weigh half gram, that is 0.5 gram, in a balance, and this powder will then be added to the buffer. Yep. Okay, it's gone above. So you can take some material out. Okay, this is 0.59. Take a little bit more out of this. Yeah, it's okay. 0.517 is okay. 18 milligram difference doesn't make any difference in this case in making the gel. Okay, so she has added agarose to 1x TAE, which is the buffer which is used for running the gels, agarose gels. Okay, you can see the turbidity. It means that this agarose does not dissolve into the buffer. You have to heat it in a microwave and boil it for 1 or 2 minutes until it is completely dissolved. Can you do it? Agarose has been dissolved in this liquid and under the microwave, and it's ready for pouring because it has been cooled down to 50 degrees centigrade or slightly lower than that. Ruj, you can start pouring it into it, and I will show this. You have added ethidium bromide into the gel, and that ethidium bromide will intercalate between the bases of the nucleotides, and the DNA will be visible. So where is the tritium bromide? I want to show actually that too. So it is here. So she just used, how many microliters did you use?
8 microliters.
Okay. Okay, so she used a few microliters of that in pouring into the gel. And now we will wait for the solidification of this gel, which will take about 10 to 15 minutes for complete drying, complete solidification. And you can see that the comb is very well immersed into the agarose gel. And when we will take out the comb, the wells will provide the space for loading of the samples, that is the plasmid sample that we will be loading here. We can see that the gel has been solidified, and you can see that gel due to its opacity, which is now different from the transparency. And if you look at from the sides, you can see that the tray is having about 1 centimeter thick layer of the gel. And you can also watch that the comb is inserted in there very well. So what we will do now is that we will take out this gel, we will release the stoppers at the end of the cassette. So one of the stopper is removed. You can see the gel very clearly now from both ends, from this side as well and from the other side. And you can see the comb. Can you show the other side? Yeah, you can see the comb is down there in the gel. And she can take this comb out outside the gel buffer chamber, or she can put the buffer and then take out the comb. You can do it both ways, but I would recommend that first add the buffer so that you can easily slide out the comb from the gel without breaking those wells. Okay, so the buffer should be slightly above the surface of the gel. So she can now simply slide out this. Yeah, and I hope if I put this stopper below the gel, you can now see actually the wells here, right here. Yes, so you can see this is the line of wells, this one, and the wells, they are slightly visible. I will try to show you again, putting some paper down there under this gel electrophoresis system. And we are going to load our plasmid samples in these wells shortly. Okay, we have placed a white paper below the gel electrophoresis system and the stopper below those wells, and I will try to show you those wells here, which I hope will be visible. So yes, you can see the wells here now. They are quite visible, and those wells will be used for the loading purposes. Yeah, you can see those wells now very clearly. 1, 2, 3, 4, 5, 6, 7, and 8 wells. So this is an 8-well comb. So you have 8 wells, and these wells will now be loaded with the samples. Now we are ready to prepare actually the samples for loading. And what we will do is that we have the plasmid in a tube, and now in order to load it onto the gel, we will take one append-of tube, and to this append-of tube we will add double distilled water, that is about 15 microliter. Okay, and we will add 1 or 2 microliters of the plasmid to it. And this mixture has the same density as that of the 1XTAE. So in order to settle this mixture down into the well, we need a high density solution of sucrose or phycol or glycerol. So the loading dye is added to this mixture for that purpose, to increase the density of the DNA so that it can settle down in the bottom of the tube. Now about 2 to 3 microliter of this dye material, they are enough for putting into this plasmid solution that is now ready for loading. And you can see that it is now a mixture of the plasmid plus water plus dye. And this is the thing that we will load on each of the wells for each of the sample that we are preparing. Ready for loading these samples onto the agarose gel. So this is a mini system, and the voltage that we calculate for any of the gel electrophoresis, horizontal gel electrophoresis system, is that we measure the distance between the two electrodes. One of the electrode can be seen down here at the bottom of this, gel electrophoresis system, while the other electrode is close to this place, which is down there under the buffer and in this lane. Okay, so in this lane where I am touching my finger. So we measure the distance from here up till here, and then we multiply it with 3. Okay, and that is the voltage that is required to run the gel. This is how we calculate it. Okay, so she is now ready to load the gel, and we have set the power supply to 60 volts. That is calculated according to the formula I just told you, the distance between the two electrodes multiplied by 3. Okay, and let's load the gel now. So the 2 of the samples have been prepared, and she is taking about 15 microliter out of 20, and you can see that those samples are going down the well very well. They have settled down due to their higher density, and this is a perfect loading. There is no outflow of the dye or the DNA solution, and it is a perfect loading. You can put the other sample too. In the second well, again you can see that this sample has also been settled down very well in the well. You can clearly see it because we had 2 samples. Okay, one of the sample indicates the marker, DNA marker, and the other one is the isolated plasmid, right? So we will run this at 60 volts. Can you set up that lid over that apparatus? We put the lid red over red and black over black. The wells should be close to the black electrode. Okay, because DNA is negatively charged, it will move from black electrode to the red electrode. Okay, so we'll see that migration in a while. So she has just hit the run, and now this equipment is running at 59 or 60 volts. What you can see is that as soon as you hit the current, you can see the bubbling. This is what? It is hydrogen which is coming out of these electrodes. Okay, and because the formula of water is H2O, so 2 molecules of hydrogen, 1 molecule of oxygen. Oxygen is coming out of the other electrode, which is on this side. I don't know, we can see that, or no, not this, from this side. Okay, let's try it from the other side. If we can visualize, the oxygen is coming out at a very slow pace. Yes, I can see it. I can see it actually. Yeah, you can see those bubbles coming out of the electrode right from there below my finger, okay, on this area. But because oxygen is one molecule, hydrogen are two molecules. So you can see that hydrogen is coming up very fast from that electrode. Okay, so this is an indication that your gel electrophoresis system is running perfectly as desired, and you can see actually the migration of dye has also started. This is the indication the DNA is also moving within the system. Okay, so in a few minutes you can see this will go down towards the other pole, and we will run this system for about 30 minutes, and that is the enough time to resolve the DNA in this agarose gel. Okay, we'll keep on monitoring for 30 minutes, okay, and we will check how the migration of this dye over time, and then we will visualize the gel under the photo documentation system, and we will get a picture and will show you that how the DNA appears on the gel. It's now 15 minutes that the system is running, and we can see that the dye is moving ahead. It's not that clear because the vapors, the gases which are evaporating from the buffer, they are carrying some water molecules too, and they are making this VN a little bit hazy. What I will do is that I will stop this gel by clicking on the stop button, okay, and I will take out this lid from here. I will just pull this out, and we will see that how far this gel has run. Now you can very clearly see actually the wells as well as the dye is traveling towards the red electrode, which is the minus. You can see the wells. You can see that from each of the wells that we loaded, there are two types of dyes coming out. One of them, which is moving ahead, is bromophenol blue, and the other one is xylene cyanol FF. So the loading dye is actually a composition of glycerol or sucrose plus two dyes and plus some water, and sometimes we can add some light buffer in it, like tris, 1 millimolar tris to it, and the mobility is quite clear. From the wells, this sample has come down. The front dye usually moves with 500 base pair, and the second dye moves at about 2.5 kb positions. So we can have actually a track of the movement of DNA with the movement of dye. And this gel is run just about 15 minutes, and if we run about 30 minutes, 15 more minutes, then we can easily see the different types of plasmid configurations, that is supercoiled, linear, circular forms of the plasmid, because during the isolation process, due to the physical forces, the supercoiled plasmid gets some nicks, or it gets a double-stranded DNA break, and it's visible on the gel in three different forms that we have already talked in our class. So this is how the gel runs actually, and what we will do is that we will see this gel under a UV light in about 15 to 20 minutes, and I will take actually the picture from there, because we don't expose ourselves to UV radiation to protect our eyes, and I will actually show the picture of the gel after this gel is run properly for 15 or 20 more minutes. This is UVP photo documentation system, and we bring our gels here to see the DNA bands on it. And this is a little bit overview of this instrument. If I open the door, you can see a UV transilluminator there. And we place our gels here before we wash them, and I will show it to you shortly. Can you put the gel on this transilluminator? Yep, this is the gel that we ran for the plasmid isolation. We can watch actually this gel inside the equipment. So you can see that the DNA is visible as orange fluorescence, and we can take the picture on the monitor here. So you can see it on the monitor. So the plasmid DNA is visible as a single band. This is run for about 30 minutes. And if we run it more, we can resolve the bladder, which is on the left-hand side, to see the exact size of the plasmid, which is actually in the supercoiled form or in the linear or circular forms. They will be visible later on.